Okay, today on the Arabidopsis Research Roundup, I'm talking to Alastair McCormick from University of Edinburgh, and he's going to discuss briefly his new paper in Plant Biotechnology Journal with the title Introducing an Algal Carbon Concentrating Mechanism into Our Higher Plants, Location and Incorporation of Key Components. So, hi Alastair. Hi there. Can you give us a quick summary of the paper, first of all, please?
Sure, I'll try to be brief. So the paper basically discusses the first steps we've taken to try to introduce the algal carbon concentrating mechanism into higher plants. So basically as background, plants, specifically C3 crops like wheat and rice are considered photosynthetically inefficient because of the shortcomings of the enzyme ribisco. So ribisco is the main enzyme that plants use to take up carbon dioxide, and because it evolved several million years ago when oxygen levels were low, it unfortunately struggles nowadays when oxygen levels are relatively higher, such that it can't really distinguish between oxygen and CO2 as a substrate. So unfortunately, in our modern world, ribisco performs a lot of wasteful oxygenase reactions, which basically impact on plant performance. So what algae do in contrast to that is that they have this highly efficient mechanism called a carbon concentrating mechanism, which allows them to actively pump inorganic carbon into the chloroplast. Importantly for us, this all happens in a single cell. So what we've really focused on in this study was to try to express the known genes of the algal carbon concentrating mechanism into the mesophyll cells of plants and to test what they do. So I suppose basically what we found, we were pleasantly surprised to find, was that most of the components localized to the same place in higher plants, specifically in tobacco when we tested it, as in algae. And we borrowed these genes from the model algal species Chlamydomonas. So we also examined the kinetic activities of some of the putative inorganic carbon pumps, which are the transmembrane proteins involved in actively taking up inorganic carbon. And we found that they worked outside of Chlamydomonas, which is good evidence that they could function in a higher plant system. We then expressed these stably in another plant, Arabidopsis, and found that there were no deleterious effects on growth, which is quite exciting news going forward. And that's pretty much the paper.
Okay, excellent. So a paper with real potential down the line, translational potential, let's say. So what sort of techniques did you use for the majority of the experiments you did in the paper?
So we did a variety of things. We did a lot of cloning, of course, to organize the genes into the appropriate expression vectors. We then did transit expression and localization studies in tobacco. We did 14C uptake assays in Xenopus oocytes, for testing the activity of the bicarbonate pumps. We then did stable transformations in Arabidopsis, Westerners to check the presence of the protein, some photosynthetic gas exchange to measure the photosynthetic activity of those plants, and of course the growth analysis associated with them.
Okay, so one thing stuck out to me there, I guess, about using the Xenopus system. So why did you choose to use that? It's obviously slightly different to any plant system. So
it was a convenient system to do some assays to measure the kinetics of 14C uptake, or at least bicarbonate uptake. So doing this in higher plants is slightly more difficult, whereas Xenopus oocytes would give you quite a nice simple cell system to test heterodicy, whether these pumps are active outside of Chlamydomonas. Okay. So basically, it was just a convenient choice.
Fair enough. And so, final question. As I said, this has got great translational potential, and many people are trying to improve photosynthesis in many ways. So, where do you see the experiments that you've achieved in this paper? Where are you going with them now in future work?
Well, I suppose the main thing that we showed from this work is that the algal carbon concentrating mechanism components that we tested appear to all localize by and large, the right places in higher plants as you would find them in algae, which is really great news going forward. It seems that the systems are quite compatible. So in order to actually get improvements in photosynthetic activity, what we're planning to do is to stack several components together. The main idea is we need to identify the minimal set of components that are required to achieve a similar concentrating mechanism you find in Chlamydomonas, and then to test, of course, whether those components actually work outside of Chlamydomonas. The other thing which we're doing at the moment, which isn't mentioned too much in the paper, but it's sort of alluded to, is that we need to get ribisco aggregated together in a specialized structure known as a peronoid if we're going to duplicate the algal carbon concentrating mechanism. So this is really a structure which consists primarily of ribisco aggregated together in a sort of a ball-like structure inside the chloroplast. So we're basically working now on mechanisms to get ribosco to aggregate. And luckily, our collaborator, Martin Jannicus in the Carnegie Institute, has identified several new components which are involved in that aggregation effect. And we're working with him at the moment to introduce those into a high-blast system.
Okay, that's interesting. So I've been listening to some talks recently about bacterial carboxysomes. So it's kind of the same idea that algae, bring their rubisco all together in order to get higher efficiency, I guess.
Exactly right. Very akin to a carboxysome. Okay. Slightly more dynamic, but very similar in, I suppose, what they do.
Okay, excellent. Well, we look forward to hearing more about this over the coming years, I guess. So
thanks very much.
Thanks very much, Alastair.